Except for laboratory experiments, most microbial life takes place in complex microbial communities composed of many interacting species. Microbial communities are abundant in the soil and in the oceans, where they participate in the global cycling of oxygen, carbon, and nitrogen. They're abundant on and within our body, where they contribute to the balance between health and disease, for example, by preventing the rise of pathogens. This is why being able to predict and control the dynamics of these communities can have enormous value in environmental sciences and biomedicine. My name is Daniel Segre. I'm an associate professor at Boston University. The work that we're presenting in the latest issue of Cell Reports is focused on understanding the flow of molecules and energy in microbial communities. Like the dynamics of these communities, also our work is the collective outcome of the interaction among several researchers with different backgrounds: microbiology, physics, computer science, biochemistry. One of the fundamental ways in which microbes interact with each other and with the environment is through their metabolism. For example, two bacteria next to each other might fight for the same nutrients, or they may help each other. What one microbe discards as a byproduct, the other may use as a nutrient. Nobody really knows how frequent these interactions are in natural microbial communities and how they collectively affect the fate of an ecosystem. We wanted to know whether it is possible to use mathematical models and computer simulations to predict and perhaps engineer these interactions and to understand how they affect the community dynamics. We developed a computer program that we call COMETS, Computation of Microbial Ecosystems in Time and Space. At any given time and location, and for each microbial species, COMETS computes the internal molecular budget of the cell involving thousands of reactions and predicts how fast that microbe can grow and what molecules are used or dumped into the environment. The molecules available in the environment can then spread due to diffusion, mediating interactions between different colonies. To test whether our simulations really work, we used small communities for which we could perform experimental measurements in the lab. We first studied an engineered cross-feeding community in which two well-known bacteria, E. coli and Salmonella, give to each other essential nutrients. It worked. How about three species? We engineered a new three-species community, and it worked again. Many other microbial species can be added to these simulations, ultimately based on the availability of their genomic sequences. One of the interesting things we learned by developing COMETS is that the spatial organization can have unexpected consequences on how microbes interact metabolically with each other. Imagine, for example, that you are a bacterial colony exchanging food with another friendly colony over a given distance. Here comes a friend of your friend that gets in between you and your friend, effectively becoming a competitor of yours, stealing all the food originally meant for you. One might expect that this shadowing effect, a sort of metabolic eclipse, will make you starve. But COMET simulations for the cross-feeding E. coli and Salmonella pair predicted the opposite outcome. As strange as it may sound, it turns out that the competitor cutting between you and your partner ends up providing you with a net benefit because it feeds your partner so well. The experiments confirm this prediction. Because of the complexity of microbial interactions, tools like COMETS are going to be useful for many applications, for example, for guiding the design of artificial microbial communities as sources of renewable energy, or to help understand what combinations of friendly symbiotic bacteria are most successful at outcompeting pathogens. In addition to the fascinating challenge of describing an ecosystem starting from the genes of its constituent organisms, the prospect of a quantitative, predictive understanding of microbial communities could transform the way we approach these problems.